We're going to finish this set of notes today. I'm not sure if we'll get finished with all of the brain notes for your test. We'll have to see. Maybe we will. If we don't, I'll just tell you where I cut it off and roll over some material. I don't really like to rush through the brain, so if I do slightly less but a little better, it's better for me. So, but at least this set of notes will be on your test, and the vast majority of the next set of notes will be on your test too. But hopefully you're starting to get this one anyway, right? It's starting to stick in there, I hope. I do want you to remember that although you want to rest right after your lab practical, lab is 20%, lecture is 80%. So you need to get back in the saddle and get going on studying for your lecture test, right? And that means that that lab practical that you just took was less than 10% of your grade for this class. So, because it was less than half of 20%, right? So you need to get back on and studying, and every single lecture test is 20% of your grade. So go ahead and start working on this one, please. I want you to do well. Okay, so the rest of this set of notes, we're going to look at how neurons are put together, what they do, and then we'll talk about regeneration, repair, and the nervous system. Ask as many questions as you want. I want to make sure that this stuff gets in really well before we start the other set of notes. So we'll start easy. This stuff right here, you kind of did in the last, in the histology notes, when you just sort of looked at the structure of a neuron. So we're just going to do it in slightly more detail, and then we'll move off from there. So first thing to talk about is that when you look at this neuron, you should be able to tell me what structural category of neuron it is. Structural. Yeah, it's multipolar. And usually when you learn basic neuron structure, you look at a multipolar neuron because A, they're easier to understand, and B, they are the most common. So that's what we're going to look at today, is sort of the structure of a multipolar neuron. Okay, so it's not outrageously hard, just a few things to add to it. First off, this is the neuron cell body, right, also called the soma. What is in there is almost all of the organelles, you know, the nucleus and the nucleolus and the rough ER and the smooth ER. And so when you look at an actual histology slide, remember that neurons tend to look like owli, the owli nucleus, because the nucleolus tends to stain really dark and the nucleus is slightly lighter. And just as far as histology goes, usually the cytoplasm looks like it has kind of a granular appearance, because those things that you're seeing are they're called Nissl bodies, or the chromatophilic substance, but what they really are is the rough ER, because the vast majority of neurotransmitters are amino acid or peptide based. And so this is going to be really active in a neuron. The rough ER is going to be kind of abundant and really active. And does that make sense? Yeah? Okay, so the cell body is going to have some processes sticking off of it, and we already learned these. The dendrites are the shorter and more abundant of the two, and the axon is the longer, although I mentioned this to you last time. On a histology slide, I do not think it's really possible for you to tell axons from dendrites. This one, of course, does look longer, but you can't really tell how long any of them are, so it's a bit of a fib to say axons and dendrites. However, if you have a drawing, the axons are never myelinated, and that's this sort of insulating coating. And did I say axons? The dendrites are never myelinated. The axons is myelinated, or at least potentially myelinated. And there's only ever going to be one axon, and there can be multiple dendrites. So we'll come back to that in just a second. Now, the neuron cell bodies, I need to tell you, put this back to the whole gray matter, white matter story that we talked about before. The neuron cell bodies, when they are collected and when they are concentrated, they tend to make the tissue gray. Okay, and so in the central nervous system, especially in the brain, you're going to find collections of neuron cell bodies, two big places. First, sort of spread evenly across the surface in the structure called the cerebral cortex. And the cerebral cortex is about 2 to 4 millimeters thick all along the surface of the cerebrum. There's also a cerebellar cortex, which is also made of gray matter. But in addition to that, I want to tell you that when it is deep within the brain, you can still have gray matter, right? But that's not the cortex. Those are called cerebral nuclei. When they're within there, they used to call them, and you'll still see them referred to in like popular literature as the basal ganglia. but that's actually an incorrect association because a ganglion is peripheral and not central. Okay, so these are the parts. There are paired chunks of gray matter within the central nervous system instead of on the surface of the central nervous system. So these are called nuclei, okay? Singular is nucleus. And when we get into the brain structure functions, we'll start learning a little bit more about what the nuclei do. For instance, in here, like the amygdala, have you heard of the amygdala? Like your rage center, yeah, those are in there. You've got the nucleus accumbens, which you can't see in this one, which is a little further anterior. That's like addiction. A lot of your like emotional pathways are like in the middle there. So do you understand what I mean? So if you got chunks of gray matter on the surface, it's cortex, and deep, it's actually going to be nuclei. So this is incorrectly labeled. I just love the figure, so I'm going to use it anyway. Okay, now if they are neuron cell bodies outside of the central nervous system, you call them ganglia. Singular is ganglion. So remember these little lumps that you can find just lateral to the spinal cord? That's an example of a ganglion. So that's the correct usage of the term ganglion. And what I mean is what's inside there is neuron cell bodies. Okay, you don't have very many neuron cell bodies outside of the central nervous system, but when you do, they're in little blobs called ganglia. And we'll learn more about their function later. Okay, so when we look at a neuron, let's look at the processes of a neuron and put this together with cell biology and then move forward so that we can start talking about the cell biology, anatomy, physiology connection. First off, you have always heard that dendrites are for receiving information, right? And axons are for sending information, but that's directly related to some stuff that you learned in cell biology. Remember learning about protein receptors, cell membrane receptors in cell biology? Can you see how if these have abundant cell membrane receptors, that they would be receiving information? Yeah, that's what it's all about. They are actually receiving information in the form of chemicals, right? Like chemicals are bonding to them and actually causing a response. So these are receiving information from, for instance, other cells. And then this part right here, this axon, and specifically this part right here, has the capacity to actually take whatever information came into the cell body and make it into an electrical stimulus that goes 250 miles an hour fast down the axon. So it's going to send information unidirectionally away from the cell body. So do you see that that's like receiving cell body sort of collects it and then sends it out via the axon? Is that okay? So this part right here is physiologically really important. It's called the axon hillock. It's the part that actually can make it into an electrical stimulus. Okay, so with the axon, there only is ever one. And what the axon does is carries information away from the cell body. Now, even though there's only ever one, it doesn't mean it can't branch. It means that there are never two attached to the cell body. So there's no limit on how many times it can branch. When it branches in the middle, it's called an axon collateral, and sometimes they do that, but all of them branch at the end. Do you see the end down there? Okay, so I want you to imagine that this is the cell body, this is the axon, right? Okay, and then they always branch at the end. The branches at the end are called axon telodendria. Telodendria. Telo means end, and dendro means fingers, so it's literally the fingers at the end. Okay. And then when you get to the very end, notice that the fingertips don't look like mine. They look like ET fingertips. Do you see them? Little bulbs on the end? Okay. And those are called synaptic knobs. Unfortunately, nobody can agree what to call them. Your anatomy book calls them synaptic knobs. Your physiology book, if we're still using the same one here by the time you take physiology, calls them axon terminals or synaptic terminals or sometimes terminal boutons. But the reason I'm telling you that is I'm not going to use a bunch of terms like to trick you, but I want you to understand that the things at the end are physiologically important because they store and release neurotransmitters regardless of what you call them. So even if you run into a different term later, you're still going to understand, yeah, it's fine. Whatever you want to call them is cool. I'll remember it if you want me to. But what they do is store and release neurotransmitters. That's why they're so bulbous looking, because they've got neurotransmitters stored at the end. Go ahead.
So there's one axon.
One axon, uh-huh.
So then what about like bipolar?
Oh, that's a good question. Okay, so let's go back. Got this figure. So the weird thing about bipolar, let me see the figure from your textbook, because I think, no, this one's fine. Okay, so the deal with like a bipolar one is that although you can't tell by looking at it, this side would be dendrites leading, okay, and then this side would be the axon, right, with the bipolar one, okay. And why would you know the difference? This one would have abundant receptors and this one would be myelinated. Okay, so you'd be able to tell the difference, for instance. But with this one, it's even more confusing because with this one, what you have is dendrites on this end, and then the axon starts right there. There's still only ever one. It's just that it bypasses the neuron on its way by. And instead of like using the neuron cell body to like gather everything, it's just like, I already made the decision and just like wave as you go by. So the axons are connected directly to the dendrite. And that's part of the reason they don't usually teach structure-function relationships with these, because they're way more confusing to understand. It's really easier when you think about the neuron cell body as like gathering up everything and then sending it off down the axon. That's not the only way it occurs. It's just the most common way that it occurs. That's actually a good question. Okay, so we have the axon hillock is where it starts, the axon telodendria are at the end, and then at the very end, the tips are the synaptic knobs, and we'll talk about their function in just a second. So what happens is these synaptic knobs are going to release neurotransmitters to communicate maybe with another neuron. Could also be a muscle cell, but maybe with another neuron at a location called a synapse, and I'll teach you guys about a synapse in just a second. Okay, so before we get to the synapse, let's back up and talk about this myelin sheath. So the myelin sheath is insulating the axon. And they always say that it speeds up transmission down the axon, but we will see that that's kind of true, but a little oversimplified. What's going to happen is, here's a good metaphor for you. We have an electrical stimulus called an action potential, and we need for it to go from the axon hillock all the way to the end of the axon, okay? And I want you to imagine doing a science fair project in which you are trying to get an electrical stimulus to go from the battery to the light bulb, right? And you're going to use a copper wire between them, right? And if the copper wire is short, you don't need to insulate it, correct? Right? It's short and it doesn't touch anything on the way from point A to point B, you don't need to insulate it. If, however, you need for the copper wire to go through your walls and actually light up a light bulb, you're going to have to insulate it for a couple of reasons. First off, and it'll burn down your house, but we don't have to worry about that in the body. But second, anytime it touches something, it can actually lose the electrical stimulus, right? So the longer you get, the more you need myelination. If two cells are right next to each other, you don't necessarily need to myelinate the axon between them. But remember that the longest axons we have are the ones that go from, for instance, the cell body in the lumbar region of my spinal cord all the way down to my little toe. And if you don't myelinate those, the impulse is not going to get from point A to point B. So myelination is sort of this insulation. It will also speed up the transmission. But in addition, you can say speed up, but if you pull off the myelination, it won't get through any more than that copper wire would get through, feet of transmission in your wall. So it's not just speeding it up, it's also making it possible. Okay, and we'll talk about how that occurs in a second. Okay, so myelination, notice what it looks like is that you have like sausage links that go along the axon with little bare places between them. Do you see that? Okay, so let's talk about how myelination occurs. The myelin typically is going to be white in color. It's made primarily of fats. It's made primarily of phospholipids because what it really is phospholipid bilayer that sort of donates itself to protecting an axon. So let's look at how it occurs. This is one of the ways that myelination can occur. So let's take a cross-section of this axon and look how myelination occurs. So here's the axon. Let's say that the neuron cell body is up there somewhere, right? And the synaptic knobs are down there somewhere. And I'm going to try to myelinate. What happens is this other cell, not a neuron, this is a type of glial cell called a neurolemocyte or a Schwann cell. The Schwann cell is going to devote its entire self, its entire life, to myelinating. So watch, this is step 1, and then step 2, and then watch step 3 and 4. It's going to continue to wrap and wrap and wrap and wrap, so that mostly what you have is layer after layer after layer after layer of phospholipids, right? on top, on top, on top. And almost all of the cytoplasm and the nucleus gets pushed off to the side. So it's really like devoting its entire self and its entire like cell to the myelination process. And why does it end up being white? Well, a single phospholipid membrane, phospholipid bilayer, is going to be clear, but so is a single layer of saran wrap. And after you start putting layer after layer after layer after layer, is the whole roll clear or is it opaque? Right? It's no longer translucent, it's opaque at that point. It's opaque and kind of light in color and white in color. So what happens is when you get enough of this myelination collected, it tends to make the tissue look white. So back to our figure again. So we talked about gray matter and now let's talk about white matter again. So when I have a bunch of cables of myelinated axons that go between the regions of the brain, it tends to make the tissue look white. So what you're seeing here is mostly very few cell bodies of neurons and almost all just myelinated axons going between cell bodies. And one more time, the gray matter is not so much gray because it is cell bodies as it is gray because it doesn't have very much myelin, right? It's just a bunch of cells that are right next to one another, so you don't really need very much myelin to get them to communicate with one another. You got it? So that's why gray matter and white matter have these sort of arrangements within the brain. And generally, between any two chunks of gray matter, if you get them a little further apart, they're going to need white matter between them, right? So how do I communicate between here and here? Oh, I need white matter. How do I communicate from the cerebral cortex in the anterior portion of my right cerebral hemisphere to the posterior portion? I have to have white matter that goes all the way. Does that make sense? Okay, now let's go back to the peripheral nervous system. Remember, the peripheral nervous system is very few cell bodies at all, so almost all of it is white. Yeah? It's myelinated axons over and over and over again. It's not just myelinated axons, they're also wrapped in layers of connective tissue, right? But a lot of your connective tissue is white as well. So when you're looking at a cadaver and you see this white tissue, right, maybe running parallel to a nerve or parallel to an artery. If it is white, and if you can roll it in your fingers, and that'll start to be important when you guys start working on the nervous system versus arteries versus veins. If you can roll it in your fingers and it's not hollow, and it's running parallel to two tubes, then it's going to be the nerve. And then the tube that stands up is going to be the artery, and then the tube that collapses is going to be the vein. So, and a lot of times they run together, so you need to be able to tell them one from the other. And later on I can do that visually, right? But when you're first getting used to it, a lot of times you have to do it like tactile. You're like, oh, I have to touch it to figure out whether it's an artery or vein. Because arteries and veins are hollow and nerves are not hollow. Matter of fact, when you rub them between your fingers, you can almost feel like fibers within them. It feels kind of like a soft coaxial cable that you can feel the fibers in there. That makes sense? So almost all of your peripheral nervous system tissue is going to be white, right? And within the CNS, you're going to have white and gray. Yeah? And that's simply because you got cell bodies. Okay. So now there's actually two different ways of myelinating an axon. The one that we looked at was the way that it occurs. This is the way that it occurs in the peripheral nervous system, because this little cell called a Schwann cell or neurolemocyte devotes its entire self to wrapping around the axon. But the way that it occurs in the central nervous system is not like this. This is in the PNS. In the central nervous system, it's like this. Notice that it accomplishes the same thing on the axon. Do you see that? But it does it by a different mechanism. So instead of this neurolemocyte wrapping itself around, like individually a little chunk and making like sausage links, what this one does is the cell body of this, it's a different kind of glial cell. These are both glial cells, but this is a different kind of glial cell. And the CNS, the cell body sits off separate, and it wraps fingers around little sections of axons, okay? Fingers of cell membrane around little sections of axons. So this one is called an oligodendrocyte. Oligo means many, dendro means fingered, right? And cyte means cell, so it's a many-fingered cell. That makes sense? So who cares how it occurs? Clinically, it makes a difference, because if you damage this one, then the neurolemocytes in the peripheral nervous system will do their very best to help with regeneration. They'll make a little guiding tube for the axon to try to hit, and we'll talk about that at the end of today. But these, if you damage the oligodendrocytes, go, I'm out. And then they allow scar tissue to fill the area instead of helping. That's one of the other reasons that damaging the CNS is worse than damaging the PNS. Because if you damage the PNS, the white matter in the PNS, A, you don't damage any cell bodies, but then B, the myelinating cells are going to try to help with regeneration. It doesn't always work, but they try. If you damage gray matter in the CNS, you're screwed. We all know that. But if you damage white matter, you're also probably screwed because they don't help. They like get out, and then scar tissue fills the area. And so that's the last thing we're going to talk about today is nervous system damage and regeneration. Okay, so the myelin sheath, what it's going to do is going to speed up and help transmission of action potential. So let's go back to this figure real quick. So what I'm going to have is these regions, and what it's going to do is to allow for transmission of an electrical stimulus to go faster. And so notice you have these little bare spaces between your sausage links, right? Those little areas are called nodes. It always has the word nodes in it. People still can't agree whether to call it a neurofibril node or a node of Ranvier, which is the guy who found them. Again, it really doesn't matter because if it's got the word node in it and it's referring to an axon, that's what it's talking about. Okay, so part of the problem that you guys are specifically running into is that anatomical terminology is trying very hard to switch away from using proper names. So we don't use Ranvier, we're not supposed to use Schwann, but physiologists still use them. So, and of course the medical professionals that you are going to end up dealing with are going to still use them because they were not trained yesterday. So it's an unfortunate truth for you guys that you have to learn both. So you have to learn sort of both. And I don't usually test you hard on both. I just don't want you to be confused when you run into something you think I know nothing about that because the term is unfamiliar. I want you to think I do know about that and I can learn this new term, right? Okay, so now I'm going to tell you a short version of this physiology, and it's a little too simplistic to be really truthful. But a lot of people ask, what are these things for? And what they are for is, okay, so the electrical stimulus needs to go all the way down this. And if it were unmyelinated, which the short ones are, it would go continuously all the way down, just like it would like an uninsulated copper wire. It just sort of goes continuously down. But when it's myelinated, it ends up speeding up the transmission and making it go very quickly down long axons, because what happens is the electrical signal goes here, and then you don't let yourself lose the electrical signal, and then it goes here, and then it goes here. So instead of going continuously, it actually jumps from one of those little bare spaces called nodes to the other, which increases the speed of transmission like fourfold. However, it also means that the areas that are under the myelination don't have the ability to allow for a continuous transmission. So if you demyelinate a myelinated axon, it doesn't work at all. Okay, so this type of conduction in which you go, instead of continuously, you jump from one node to the other, is called saltatory conduction. Because I don't know if you speak Spanish, if anybody does, saltar means to jump, right? So it's this jumping transmission. And it's pretty hard to explain in physiology, but that'll like get you started. So basically, instead of going continuously, you jump from node to node. Okay, so that's what the nodes are for. Okay, so now you have a lot of information, and so what I'm hoping for with you is that now with this background in anatomy, you can look at any individual neuron, especially a multipolar one where it's a little easier, and tell several different things. First off, you can tell direction of transmission, right, very easily. You can label axons versus dendrites, yeah. You can label the axon hillock. Yeah, the axon hillock, which is here. The soma, or cell body, yeah. The axon telodendria and synaptic knobs. You could also, and this is probably the harder part of it, could you tell me whether this axon runs through the PNS or the CNS by looking at it? Yeah, it's the PNS, remember, because the nuclei are associated with the myelination as opposed to being separate, right? That makes sense. So that the idea of getting a really strong anatomy foundation is that then the physiology ends up falling into place instead of rote memorization, instead of having to stare at it and try to figure out what's going on. And that's what I'm trying to accomplish with you guys. Okay, so now, when you get to the end right here at these synaptic knobs or axon terminals, you're going to form a synapse. And a synapse is the site of communication between a neuron and the next cell. So here's the idea of a synapse. And now, because of the information you have, you can look at two neurons in series and tell me which direction information is going between them, right? Okay, and if you had to, could you look at these two neurons and label them presynaptic and postsynaptic? Yeah, you could, right? Because you know that per synapse, this one's number 1, pre, and this one's number 2, post, right? That's for those synapses, right? Of course, this one would be pre for the next one, but you don't label presynaptic and postsynaptic globally. You label them with reference to a synapse. Is that okay? Okay, so now you could also label axons and dendrites, right? And you could do all of that here as well, right? You can label presynaptic, postsynaptic, axons, dendrites. And so what you're going to have is the site of communication is going to be the synapse. And if you blow up a synapse and look at like a single one of these synapses, this is where the really interesting physiology comes in, at least the physiology that I'm totally fascinated with. Neurophysiology is my particular love that I read just for kicks and giggles all the time. And love to study. So if I blow one of these up, I'm not going to hold you responsible for this physiology. It's just a preview of things to come. So if you go down to the end, right, you have, just orient yourself. This is your axon telodendria, and this is your synaptic knob, right? And this is all bulbous because it's storing neurotransmitters. So the interesting action that will happen here is that you are actually going to release neurotransmitters from this site, which is an incredibly energetic process. So you're going to need a boatload of mitochondria there. They're going to be stored in vesicles, and you guys learned about vesicles in 1610. You're going to release them, and actually the presynaptic and the postsynaptic look like they're touching one another, but they don't touch one another. They're actually microscopically separated. So you have room for diffusion of these neurotransmitters across this little synaptic cleft. So whatever neurotransmitter it is, it's going to be released here, and then it's going to bind to receptors on the postsynaptic cell. Then you're going to start the whole story over again, right? It's going to bind to receptors. Let's say the receptors are going to be on the dendrites of the postsynaptic cell. And then you're going to be receiving information, and then the postsynaptic cell could get an action potential and start the whole process over again. Is that okay? And so one of the reasons that well, there are tons of reasons why I find this so fascinating. Part of the reason I find it so fascinating is that almost all of the things that you associate with being a human are because of neurotransmitter activity. So your neurotransmitters being released in the proper amounts in the proper places are the reason you're brilliant or an idiot or somewhere in between, which is what most of us are, right? It's the reason that you are crazy or sane or somewhere in between, which is what most of us are, right? It's the reason you're happy or sad or depressed or schizophrenic or all of those things. It's all because of neurotransmitter activity. In addition, the vast majority of the reason that you do anything is because you are chasing neurotransmitter activity. The reason that you eat is not because you feel like, oh, I need to eat, at least most of the time, but because it actually releases chemicals in your brain that are interpreted as pleasure. You're like, oh, that was amazing. That's the reason you don't eat broccoli for every meal, right? You're like, I know I should, right? But I'm like, I'm going to eat some chocolate right now. So you are causing the release of like some of your favorite neurotransmitters. And you have favorites, you just don't know. And your 3 favorites are dopamine, norepinephrine, and serotonin. And those 3 in exactly the proper combination will give you a feeling of pleasure, motivation, and peace all simultaneously, which is like what we would all want at every moment, right? Pleasure, motivation, and peace. And I think I've been there for maybe 5 minutes in my entire life, and which is all just perfectly, like the little Venn diagram just comes together. So fascinatingly, like this is where a lot of neurophysiology research goes on, because you, for instance, have sex for the same reason that you eat chocolate, because they release pleasure chemicals in your brain, right? You jump out of a plane for the same reason that you have sex, because they release pleasure chemicals in your brain. You do methamphetamine for the same reason that you have sex. And so the pleasure chemicals in your brain are all something that we're like chasing by one mechanism or another. And so there's a tremendous amount of research there, a tremendous amount of evolutionary biology there. And I just read about this stuff constantly, and I go to neurophysiology conferences because I'm interested in it, and I teach tons of neurophysiology in both abnormal and normal neurophysiology and drug interactions on the brain when I teach phys, because I think it's a really good way to like understand what's going on. I teach about methamphetamine and sugar and opioids and all of those kinds of things, and how, you know, ADD drugs affect the brain. So this is like, to me, the most interesting part of the whole A&P story is what happens right there. So you don't do that in anatomy. You just say, hey, it exists and it's going to come in the future. So the synapse is the junction, the site of communication between two neurons. A true synapse means it's going neuron to neuron, but just so you know, neurons can actually communicate with muscle cells, right? And they can communicate with glands as well. So if cell 1 is a neuron and cell 2, which is it isn't here, is a muscle cell, instead of being called a true synapse, it would be called a neuromuscular junction. If cell 1 was a neuron and cell 2 was a gland, it would be called a neuroglandular junction, okay? But the first cell does the same thing regardless of what the second cell is doing. So you're going to learn about those synapses later. So now the question is, when you look at one of these, can you answer all those same questions? You know, direction of information, which one's presynaptic, which one's postsynaptic, where are the axons, where are the dendrites, synaptic knobs. So you should be able to, by the time you like practice a little bit, answer all of those questions. Everybody good with that? Okay. Alrighty. So that's our intro to sort of neuron functionality. And now we're going to look at our glial cells. And the glial cells are sort of the support system of the nervous system. They're smaller, but they're way more numerous. So on average, your neurons make up about half your brain volume, and your glial cells make up about half your brain volume. But they're way tinier collectively. So there's usually about 50 times more of them. Glial cells than there are neurons. And I told you guys this when we first did neurons in histology. They named them glial cells because they thought they were just space fillers between the neurons, which got all the attention. Because we know that the neurons do the electrical activity and release the neurotransmitters. And so that's a very, very interesting part of the story. And we thought everything else was just like scaffolding that you put between neurons or space fillers. And so they named them glia, which means glue. And then they figured out, oh my gosh, that was entirely too simplistic. So the metaphor that I use for glial cells, which I've probably used with you guys before, is that the neurons are the stars of the show, right? So, but they have been movie stars since they were infants. So these are like the Olsen twins, these two right here. And they have been courted to and protected and had things done for them since their infancy. And it means that they are so specialized in their functionality that they don't know where to put gas in the car. They have no idea how to protect themselves. They don't even know how to get at their bank accounts, right? They don't know how they like their coffee. They know none of those things. And so what they end up doing is they end up depending on this huge support staff of cells that do everything for them. They have a bodyguard right here. They have a personal assistant that makes sure that like they control access to them. They have the people who are in charge of their communication, right? So let's look at the glial cells. That you have different glial cells in the CNS, which would be like when you were in LA, versus the PNS, which is when you're on location. Of course, you have a bigger support staff when you're at home. So let's talk about the support staff. Okay, so the glial cells that you have in the central nervous system are the ones that you see here. But you've already met one of them, right? So the one that's in charge of like promoting communication right here, right? This may be the publicist, right? This guy right here is the oligodendrocyte, right? And it's the one that does myelination when you are in the CNS, okay? So you already know about that one. Okay, but the other ones that you have are also really important. This guy right here is called the microglial cell. You can see why it would be called that, right? And this is your little bodyguard, only instead of having a big bruiser bodyguard, you have a little ninja bodyguard who's a total badass. These little guys right here have to really protect the neuron from pathogens because we have a thing called the blood-brain barrier, which we'll learn about in more detail in the next set of notes. And the blood-brain barrier really does not allow very many things to get out of the bloodstream into the central nervous system. Only the absolutely necessary things. Well, what if something did get out? Well, the problem is if it did get out, white blood cells can't get out. So you do not have the cops, right? You have your internal security force. So white blood cells can't make it out of the bloodstream. So if anything else sneaks out, you've got no one to help them except the microglia. You've got no one to protect you except the microglia. So these little microglial cells apparently work pretty darn well, right? Because how many brain infections have you had? None? I mean, unless like I'm supposed to be 10 IQ points smarter, and this is just like what I have left. But so apparently they work very well. They work in combination with this guy right here, which is the astrocyte, which astrocyte, in addition to sort of just providing structural stability for the neuron, it also controls access to the neuron because the astrocyte controls the blood-brain barrier. It is like the gatekeeper to the superstar, right? So the blood-brain barrier is supposed to only let things get into the central nervous system that are good or necessary to the central nervous system. And it works pretty well too, but nothing's perfect. So if something gets past, then the microglial cells. So the blood-brain barrier we're going to talk about quite a bit. The blood-brain barrier is basically the capillaries that go deep within the brain have the tightest of tight junctions in them. And so things do not get around the personal assistant. They have to go through the personal assistant, right? When it's working properly. That's what they do. Does that make sense? So these guys are going to control permeability, control access to the central nervous system. Good? And then this one, I have no good metaphor for. I can't come up with one. But let me show you what this one is. So this last one is called an ependymal cell. I'll show you where it occurs and then I'll describe what it does. Okay, do you see right here? What it's trying to show you is the cross-section includes a little chunk that actually leads into this space in the brain called the ventricle. So you're going to have the cells that are actually at the border of that space. So here's the ventricle of the brain right there, that little space, right? So you've got these cells that are lining the ventricles. Okay, those are also glial cells. Don't they kind of look like epithelial tissue? They are similar in function, but they are not derived from epithelial tissue because the brain has only brain tissue in it. It's all derived from nervous system stuff. It doesn't have any connective tissue within it, and it doesn't have any epithelial tissue. But you can see why they'll be called epi-something, right? These are ependymal cells. And the ependymal cells, let me tell you what their function is. And we're going to get into them in more detail when we talk about cerebrospinal fluid. But what they do is they actually pull from the blood and from the blood plasma all of the best stuff, including a lot of water, but also including high levels of glucose because the brain loves glucose, high levels of oxygen because the brain loves oxygen, high levels of the ions that the brain needs for transmission. And then they actually make them into cerebrospinal fluid and then dump it into the spaces of the brain. So the spaces of the brain are actually fluid-filled spaces of the brain, and they're filled with the fluid called cerebrospinal fluid that was made by these. And it was made from blood plasma. That's what it was made from. Is that okay? So these are ependymal cells, and those line the cavities of the brain called the ventricles. And you guys are going to learn about ventricles starting on Friday and then next week. What? Maybe, yeah, the chef. But they're actually like getting stuff out, though. I mean, I don't know. Eventually, I've been working on that metaphor, and I can't come up with one. Go ahead.
Are they encouraging things to come past the blood-brain barrier?
No, well, they're selecting very carefully what comes past the blood-brain barrier, so they're only selecting the best stuff. Huh?
They're just there, and if things pass by, they grab it. They're not actually like forcing stuff.
Well, these can get around the blood-brain barrier. It doesn't show here, but what happens is they actually wrap around the capillary, and they end up pulling things. They have transport proteins, for instance, for glucose on them. So they're actually transporting glucose on purpose. They leave behind all the toxins and all the waste products and just pull like the juiciest stuff and then dump it into the fluid. So the brain has like two fluid circulation processes. It of course has blood, right, running through it. But in addition, it also has cerebrospinal fluid that is running inside the cavities of the brain and then like coating the outside of the brain as well. And in both of them, you only want the best stuff, right? You want the best stuff in the blood and you want the best stuff in the cerebrospinal fluid. Okay, so the brain is an interesting little beast. The cerebrospinal fluid is kind of like bathing the outside and the inside of the brain in nutrients and also sort of protecting it. It also gives it some physical protection as well. Okay, so that's your support staff that you have in LA. But if you go on location, you don't get to bring everybody with you, right? You don't get to bring all of the people with you on location. So in the peripheral nervous system, you do not have as big a support staff. We already know that you still are going to need to do communication in the peripheral nervous system. So here are the cells that you have in the peripheral nervous system. We already know this one, right? The myelinating cell in the peripheral nervous system is the Schwann cell or neurolomocyte, right? Okay, so we already know about that one. And then you have one more. And this guy right here is the one that in a ganglion, do you remember the ganglion is when you have a lump of neuron cell bodies outside of the central nervous system, right? In a ganglion, you're going to have neurons by definition, a lump of neuron cell bodies outside the central nervous system. Before we go any further, what kind of neuron is that structurally? Do you remember the name of it? Structural category of neuron. Not bi, it's got one attachment, so it's unipolar. That one's unipolar. Okay, so this unipolar neuron, and that's what is in most ganglia, not all of them, has these cells that are surrounding it and really like getting it its coffee and restricting access and doing everything, maybe even doing your damn hair, whatever. It's like your personal assistant that you have there. This one is called a satellite cell. Can you see why it would be called a satellite cell? Because it's sort of around the outside, right? Does that make sense? Okay. So the satellite cell is just sort of this generalist protector of neuron cell bodies that are in the peripheral nervous system. Okay, go ahead.
Are they always unipolar?
No, not always unipolar. Most of the ganglia that you're going to run into this semester are unipolar, but there's some other types of ganglia. Yeah. That okay? Okay, so you only have 2 in the peripheral nervous system. You have 6 glial cells total, but 4 in the CNS and 2 in the PNS. Good? Okay, so now clinical significance to glial cells. Well, first off, glial cells, unlike neurons, glial cells retain their capacity for cell division. And that is both good and bad. It's good because if you damage them, they can at least potentially go through cell division. They don't always, but they can. And it's bad because, remember, neurons are mostly amitotic after fetal development, but glial cells are not, and that means that if a cancer originates in the nervous tissue, it is more likely to be a glioma than a neuroma. Okay? Yeah? A glioma than a neuroma, because glial cells are constantly going through cell division. So it's way more likely to lose control of something that you're doing very readily and very commonly, right? So I'm not going to hold you responsible for these names, but for instance, you have ependomas. That's one that occurs in the ependymal cells. You have microgliomas. You have astrocytomas. Does that make sense? So those are some examples of gliomas. You do have neuromas, but they're far less likely. But, you know, anything that can go wrong in a human body ultimately will at some point. It's just like statistical probabilities of how likely it is. Most neurons don't even have centrioles any longer. So cell division in them is very uncommon, but apparently there are a few that still do and could still remember or potentially go through. So, and then the other thing is, let's talk about multiple sclerosis. And multiple sclerosis is an interesting disorder because it's related to myelination. Okay, so with MS or multiple sclerosis, what you have is a person gets exposed. They think it's getting exposed to a virus, although they don't know which one. You get exposed to a virus, and then your immune system ramps up because of the viral exposure, right? You make all kinds of antibodies, and white blood cells go through cell division. And it battles off the virus potentially, but in addition, it just gets a little overexcited and starts to think cells that are not pathogens are pathogens and starts to kill them off. And with MS specifically, what it likes to kill off is the cells that do myelination. And when it does that, it ends up demyelinating axons. which a lot of people think, oh, it would just slow down transmission. What it ends up doing is stopping transmission, and stopping transmission specifically in the periphery more than centrally, because remember, the longer an axon is, the more important myelination is. So with MS, what you start having is the symptoms occur, loss of motor activity, for instance, in the periphery first, right? And then it can move up. But it doesn't kill off the axon, and it doesn't kill off the neuron. What it does is just demyelinates it. And so if it goes into remission, you can remyelinate in the peripheral nervous system pretty readily. So for instance, you could go into remission, you could have loss of motor activity in your hands, and then you go into remission and you actually regain motor activity in your hands. And then it goes to a different region of your body. And like all autoimmune disorders, there is no cure for MS. It's not inherited. You inherit a genetic predisposition to globally autoimmune disorders that you couldn't know unless you have a bunch of people in your family with them. But you don't inherit an autoimmune disorder. You just kind of, it's like you inherit a hypervigilant immune system, right? Which then can manifest maybe as MS, or maybe as lupus, or maybe as some other autoimmune disorder. Regardless, what tends to happen is this demyelination causes the nervous system to short circuit either permanently or temporarily. And we don't have cures for autoimmune disorders. Is that okay? That makes sense? Okay. Fascinatingly, I can't understand this part yet, and I don't know if it is understood. It would seem to me with the clinical manifestations of MS, which is generally peripheral, like neuropathy, loss of sensation and motor activity in the periphery, it would seem to me that most MS would be demyelination of the Schwann cells. But it seems as if MS usually occurs in the central nervous system. It usually occurs up here. So it seems as if the problem is not getting it out here, but it's getting it out here to the peripheral nervous system. It doesn't quite make sense to me yet. So I don't know if that is known and I don't know it, or if that isn't known yet, because there's a lot of things I don't know. Okay, so that's MS and gliomas. Any questions? You guys good? Okay. All right, so the last part is regeneration of nervous tissue. So let's talk about regeneration of nervous tissue. So do you ever regenerate nervous tissue? And the answer is, well, it depends. It depends on where the damage occurred. Okay. It also has depends a little bit on when the damage occurred. But generally, if I damage my central nervous system, so if I damage the gray matter in my central nervous system, in all likelihood, you're not getting any of those neurons back. Most neurons are amitotic. There are a few exceptions, but most neurons are amitotic. So if I damage the gray matter in my brain, I don't usually get most of it back. But fascinatingly, your brain has a tremendous amount of plasticity. So I want you to imagine that I damaged, and we'll learn this in the next set of notes, I damaged the gray matter in the cerebral cortex of my left temporal lobe, which is, among other things, related to speech production. Okay, so I had a stroke, and I lost that. Well, here's what happens. Your cells die off because of the stroke, usually because of lack of oxygen to the area. And then what happens is your microglial cells will go in and clean up the mess because you don't leave dead tissue, right? That makes sense? And your astrocytes will fill the space. Interestingly, astrocytes are, in addition to doing their blood-brain barrier function, they're also just sort of the scaffolding of the nervous system. And so they will fill the space, and you won't get any reconnection or new cells, because the astrocytes, for all practical purposes, just made a chunk of scar tissue in your brain. So you don't get that back. But of course, you guys know that sometimes you can regain functionality after a stroke. So you don't get the structure back, but your brain has a tremendous capacity for what we call neuroplasticity. And neuroplasticity means that regions of your brain, it's like they were all cross-trained, right? Everybody knows how to do everything in the gray matter. And so what can happen is if this part of my brain dies off, I can pass off functionality, I can pass off the job to another part of my brain. It's not instantaneous, and it's not guaranteed, but it's distinctly possible that you can do that. To give you an example of like an extreme example of like showing you neuroplasticity is, so if you have a person who is blind from birth, never sees, in most of us, the cerebral cortex and the occipital lobe of the brain back here is primarily related to vision interpretation, right? It's like your conscious vision right back here. And then your hearing is mostly in the sides of your brain, mostly temporal lobes, but not exclusively. Okay, but if you have a person who's blind since birth, fascinatingly, what generally happens, and there are exceptions to this, is that the temporal lobes both still do hearing, but you actually devote the entire occipital lobe to doing hearing as well. So do people who are blind since birth hear better? No. Their sensation is the same. They process it better. It's basically like instead of a single NASA computer devoted to interpreting what you take in, you've got two, right? So your hearing interpretation, your spatial orientation, all of that has way more processing power associated with it. Does that make sense? But can you see what I mean about neuroplasticity? It seems as if most of the regions of the cerebral cortex have the capacity to do what the entire cerebral cortex does. And so you can actually sort of pass off some functionality. And so that's how you can regain functionality after a stroke. You're better at doing that the younger you are. You've got more neuroplasticity the younger you are. And women are better at neuroplasticity than men. They have far more overlap of functionality, and so they tend to recover from strokes slightly better than men recover from strokes. And there's a couple of reasons for that. One of them is the like connectivity of the sides of the brain is actually better in women. So if you lose a right side function and you have better connectivity, you could actually pass it off to the left side, right? That makes sense? All right, so that's just the white matter functionality. So now let's talk about the gray matter functionality within the CNS. So if I damage gray matter in the CNS, I generally don't get it back, but I might regain, I don't get the structure back, but I could potentially regain the function. But what if I damage white matter in the CNS? If I damage white matter in the CNS, the problem is the oligodendrocytes just bug out, and the astrocytes go in and fill the space. They're like, okay, so if I damage my corpus callosum right here, oligodendrocytes are like, I'm out, and astrocytes are like, okay, I'm going to fill the space. So even if you tried to reconnect, you would actually have a physical impediment to reconnecting the sides. Does that make sense? So like I said, it's the brain's sort of equivalent of scar tissue formation. So, and I'll talk about some experimental treatments in just a second. But if it occurs in the PNS, you've got at least the possibility for regeneration. It's not a guarantee of regeneration because things happen differently in the PNS. So what I want you to imagine is that what we are going to do is the neuron cell body is going to run through a nerve, and the neuron cell body is going to be in the cervical region of my spinal cord. And the nerve that's going to get damaged is going to be maybe my radial nerve, which runs down the lateral surface of my arm and would do maybe motor innervation of the lateral side of my hand and also sensation on the lateral side of my hand. That makes sense? Okay, so if I damage that nerve, remember that I am going to probably be damaging more than one axon, correct? Okay, but let's look at it an axon at a time and see how an individual axon might have the possibility of regeneration. So this is PNS damage, do we agree? Okay, PNS damage. Okay, so what happens in the peripheral nervous system? Well, since I just damaged a nerve, I didn't damage a neuron cell body, right? It's way up here protected. So the neuron cell body is protected. So I have some kind of trauma, and let's say we were talking about a motor axon, right? And the motor axon is going to go and control maybe this activity. Okay, so this is supposed to be skeletal muscle, even though it kind of looks like smooth in the picture. I think it's supposed to be skeletal muscle. So we are going to have a motor axon, which is supposed to connect with skeletal muscle. What happens to these skeletal muscle cells if I sever this axon? What happens to them? No, they don't work. Because remember, it's skeletal muscle. Skeletal muscle doesn't do anything unless you tell every single cell what to do. So these are paralyzed, okay? So let's talk about possibilities of regeneration. So what happens in the peripheral nervous system is, best case scenario, these cells right here, which are neurolimocytes or Schwann cells, are going to be motivated by the damage to go through cell division. And what they're going to do is they're going to start to form a guiding tunnel or a growth tunnel. And this guiding tunnel is going to start to sort of get more complex and more complete. And the axon that was damaged is going to seal, and it's going to become swollen, and then eventually after it's repaired, it's actually going to start to go through growth. But what it needs to do in order to reinnervate is it needs to hit the tunnel, and the tunnel is like directing it to reinnervation, to the right cells to reinnervate. Okay, so it's going to try to direct the tunnel, direct the growth through the tunnel, and it's going to form a physical tunnel, which is like attractive, and it's like shooting at something, just like going straight down the tunnel. But in addition, they figured out recently that it also secretes chemicals that are attractive to the growing axon end. It's called chemotaxis. So it secretes chemicals that are like courting the axon into the tunnel. Does it work every time? No. But it does work sometimes. And so let's say all goes perfectly, then what happens is the remyelination is happening simultaneously to the axon growth. Okay, so you get remyelination. If it hits the right tunnel and you give it a while and all things work perfectly, then eventually you will reinnervate and you will no longer have those cells paralyzed. Okay? But, of course, when you damage a nerve, you don't just damage one axon in all likelihood. You damage more than one axon, right? So you have to do this for every single axon in the nerve. Do you think they ever miss the tunnel? Yeah. Do you think they ever hit the wrong tunnel? Yeah. So you can get weird innervation patterns that occur. So if, for instance, I damaged the radial nerve, which goes down the lateral side of my arm, if I damaged it, first off, this whole regeneration process happens relatively slowly. So it happens 1 to 5 millimeters a day. And remember how long these are. So it can take up to 2 to 3 years to figure out how much regeneration of functionality you're actually ever going to get. But let's say you do get some regeneration of functionality. So what can happen is some of the cells can hit the tube and be reinnervated properly, and others don't. That means as far as muscle activity, I may have muscle weakness instead of paralysis, because some of the cells work and don't, and the others don't, right, in that particular area. Or I could actually have it wired up incorrectly, in which my body actually thinks it's working cells over here and it's working cells over there. And then you have to sort of retrain yourself for what your muscle is actually doing. It's even more interesting when the sensation, when you damage sensory neuron and they hit the wrong tunnel going up, because then I can touch it here and feel it over here. Because they actually just hit the wrong tunnel during the whole like reinnervation and regeneration process. Isn't that kind of interesting? So usually if you damage the peripheral nervous system, a responsible physician is not going to say you will absolutely get regeneration of functionality. They'll say, you're going to have to give it a couple years, and you may get regeneration of functionality, and I can't actually tell you how much. But it's probably going to be a couple years. So what can surgeons do about this with like PNS nerve damage? Really, all they can do is to put together the nerve, right? So if you damage the radial nerve and you got like one end over here and the other end over here, none of them are going to hit the tunnels. So they just take it and they put it back together, and usually they just fix the connective tissue. But do you think they're actually trying to rearrange all like hundreds of thousands of axons? And no, they're not going to do that, right? They're going to do their best. They're like, stick it back together. I think I got the fascicles in about the right place, because you can see those. I think I got the outside of the nerve in about the right place. Do your very best, and then you just wait, and then you just like hope and see what happens. This is PNS damage, and now I want to talk about CNS damage. So if instead of damaging my radial nerve over here, I damaged my spinal cord here, even low lumbar region of the spinal cord, then my possibilities for regeneration are very, very different with CNS damage. We haven't looked at the structure of the spinal cord yet, but I'll tell you the spinal cord is primarily white matter around the outside and gray matter on the inside. But, of course, the white matter in the spinal cord is myelinated by oligodendrocytes. So if you damage it, in all likelihood, you're not going to get any regeneration. So if you damage the spinal cord here, then you don't get transmission to everything that is inferior to there, right? You don't get sensory transmission up. You don't get motor transmission down. And so you end up with paralysis of the lower limbs and the pelvic cavity. So what about regeneration? Well, pretty much no. You don't really get any regeneration of gray matter or white matter in the spinal cord, with the exception of a couple of experimental treatments. One of the experimental treatments is really interesting. It's still, last time I read, it's still in vitro, like they're still working on it in like petri dishes, right? It hasn't actually even moved to animal models yet, as far as I know. And so generally with like clinical studies, you're going to start out in the laboratory, and then you try to get approved to do them in animal studies. And then after that, you may even use a closer animal, like you may go from rats to chimps, right? And then only after that would you ever get approved for humans. You don't always go rats to chimps, and the human approval is really hard to get, do clinical studies on humans. So this one, what they have been working on in tubes and petri dishes is an experimental treatment that causes oligodendrocytes to form guiding tubes so that you can actually potentially have regeneration of white matter in the central nervous system. And they're having some preliminary success, and it will probably move on to animal models, although I haven't checked lately to see what it does. But I'll tell you that some things we have had success with, which are really interesting, and that is, I think I talked to you guys a little bit about, maybe it wasn't you, maybe it's the other class. Okay, so this is kind of a long, complex story, but it's a really interesting one. So if you transect the spinal cord, you're going to lose gray matter and white matter, right? You're going to damage both gray matter and white matter. So what we have figured out is that you can use embryonic stem cells. Embryonic stem cells are way back before differentiation. So you go sperm and egg, start to go through cell division. You go up to about 32 cell stages. We're still microscopic, and we have not done any differentiation. So this is not a baby anatomically or physiologically, because you haven't differentiated any of the cells yet. You specifically don't want differentiation because you want the cells to still be at the stage that they could make any human cell. Does that make sense? So you want them pre-differentiation. In addition, importantly, pre-differentiation, you do not have almost any capacity for rejection of tissues from other humans because they don't have any antigens yet. And the antigens are the things that the immune system recognizes. You guys with me? Okay, so embryonic stem cells can form any cell in the body. So I'll tell you about the rat study, and then I'll tell you about how it's been used in humans. There's some famous rat studies. One of them completely transected the rat spinal cord so that the rats were paraplegic. They lost sensory and motor control of their pelvis and lower limbs. And then what they did is they took rat embryonic stem cells, species specific, and before you let the astrocytes move in, and this is super important, before you let the astrocytes move in, you inject into the region the rat embryonic stem cells. And embryonic stem cells are amazing because they seem to take cues from the tissues around them as to what they should differentiate into. So they go into the area and then they start the differentiation process. And within 4 weeks, they had a 70% regeneration of functionality in rats, which is freaking amazing. It's like, it's huge, it's huge. And so the rat study was obviously, really fascinating. So how do you move this up to humans? Well, you need spinal cord trauma patients. That's what you need. But then you have to have a very specific subset because you don't do children before you've done any clinical trials, right? Even though it would, like as a parent, you think, well, try this, right? But you have to, like clinical trials on children. So you can't do children. And you have to have somebody who has no other complicating factors, right? You have to be basically accepted into the clinical trial. And then you need to remember, this needs to happen very quickly, right? So you need to, within, I think it was 72 hours, although I can't quite remember now. Within a short period of time, you need to get the person in for embryonic stem cell treatment. But spinal cord patients, they don't fly, right? And you don't want them to travel very much. So you kind of need to be close to the region of clinical trial, right? And of course, you're going to have to sign off on the clinical trial and very quickly make that decision. You're just going to have to make it super quickly. And then you can inject the embryonic stem cells into the region. And we have had regeneration of functionality, not to rat. Well, with the rat, it's like all a controlled study. You have no other damage. You like specifically did a slice, right? So, but we have had 30 and 40% regeneration of spinal cord functionality with people. We've got a much smaller group, a smaller subset, because you can't like cut somebody's spinal cord just to see what happens, right? You can't really do that. So yeah, we have a much smaller subset, but the big tragedy, and I don't know if I talked to you guys about this, is that none of this really, hardly any of this goes on in the United States anymore. And the reason is because during George Bush II's presidency, people got all confused and there was a whole bunch of misinformation about what embryonic stem cells were and what they were for, and it got all conflated with the abortion argument. So did I talk to you guys about embryonic stem cells before in this class? Okay, so I talked about them in the other class. So here's the deal with embryonic stem cells. There's tons of misinformation out there, but it's very easy to find the true scientific information. It's not even like my particular perspective couldn't be confirmed by science. So if you disagree with me, you just go figure it out. You just go like find out how I'm wrong. That's totally cool. So here's where embryonic stem cells come from. Specifically, there is international law to make sure that whatever embryonic stem cells you are using for a purpose like this have to be tracked back to a fertility treatment and an in vitro fertilization. So it's like a chain of custody, which means, and the important part is that means that I can't create them for this purpose, okay? Because you can see that people would get very disturbed about you creating human embryos for a purpose of using in another human. So they tried to take care of that. It's not to say that no one will ever do it, but it is internationally illegal, and you can't use them for research, for like funded research, unless you can track them back with the chain of custody. So let's say I'm having fertility problems, and my partner and I decide to go through in vitro fertilization. They give me drugs so that I hyperovulate, right? I ovulate more than one egg. And then they harvest my eggs, and then they use sperm from my partner, and they do fertilization, and they do it in test tubes or in petri dishes instead of in my body. So fertilization happens out there. But it's really expensive and generally not covered by insurance, and it also has a pretty low efficacy rate. I think the efficacy rate is less than 20%, although it may be up to 25% now. And it's around $10,000 to $14,000 a pop every time you do this. It's very expensive, okay? So what would happen is that understanding that efficacy rate and that cost, I would not say I want 2 children, I'm going to fertilize 2 eggs and then try 2 implantations because the efficacy rate is so low. So generally what would happen is I would try to fertilize 20-ish eggs, right? However many I can get out at the time. And then I don't stick them all up there at the same time, but usually you either do 1 or sometimes they do a couple. Every once in a while they do like 8 and then somebody ends up having octuplets, which nobody wants, right? So you stick them up there and then you just see if they actually implant. And if they implant, you get a pregnancy. And if they don't, then you just don't get a pregnancy. So let's say I go through that and I have the kids that I want. I didn't intend to have 20, even in Utah, right? I may have intended to have 3, I may have intended to have 4, it may take 6, 8 years for me to have those 3 or 4. Here's a question that people run into. What do you do with those extra 16 embryos that are frozen at the 32 cell stage so that they can't go through any further differentiation and are never going to form into a human until you thaw them and implant them, right? What do you do with them? And what most people decide is that they don't do anything with them because they can't figure out how to make the right decision that feels good to them, so they just leave them frozen. Well, if you leave them frozen for 2 years, they're no longer viable for implantation. If you leave them frozen for 5 years, they're no longer viable for anything, research or anything else. And that's the decision that most people make by default. They just don't decide what to do with them. So how does this whole story tie back to this? This whole story ties back to this and a lot of other disorders because people got this whole issue sort of confounded with the abortion issue. and people started saying that embryonic stem cells are baby parts. And so so they removed, so George Bush, being the incredibly brilliant human that he was, I'm from Texas, so I can say anything I want about him because I voted for him every time anybody would let me. I voted against him every time anybody would let me. He removed all federal funding in the United States for all federal research, which means that what happened to our embryonic stem cell researchers? They left. And so most of this research that has gone on in like Scandinavian countries and Israel and Germany and the UK, and we hardly have any access to it any longer because it's like a time and space issue. And we lost all of our researchers, and they haven't come back, right? They haven't even restored, Obama restored a bunch of the money for embryonic stem cell research, but the problem is that the research programs were already gone. And it just didn't come back after that. And it may, I mean, if you give it 20 or 30 years, it may come back, but it was an enormous blow. And the problem, fascinatingly, like this is like an editorial comment. You disagree with this as much as you want. The fascinating thing that I always ran into when I like looked at this issue was we're having the argument at the wrong place. And so let's say, just theoretically, that it is absolutely your position that life begins at fertilization. If that is absolutely your position, then that's a totally fine position for you to take. We don't have to agree, but it's a totally fine position for you to take. But if that is your position, and that is sort of what he was presenting as his position, then why are we not arguing about in vitro fertilization? Why are we not arguing about the 20 embryos that you did not intend to turn into 20 humans, right? Why are we not having that argument? Because that's a consistent argument, right? And it would be consistent with your position. But no one, even like here in Utah, where we love some babies, we were not doing anything about in vitro fertilization, but there were an awful lot of people who were very, very much against embryonic stem cell research. And there are a bunch of reasons to think about that. From my perspective, what I kept hearing and my students kept asking me, I was like, this is abortion, this is baby birth. That's a bunch of misinformation that's being put out there. I mean, you can find a reason to disagree with it from your personal perspective, but if it's based on misinformation, it's not going to hold up, right? If it's based on consistent information, that's fine. We just disagree, and we can vote, we can battle it out in the voting box. But when it's based on misinformation, like my perspective is like, you gotta actually know what's true, so you figure out whether you agree with it or not. So anyway, this is really, I mean, I know people with spinal cord injuries, and I know people who have spinal cord injuries who've had them in the last 10 years. And this directly impacts the quality of access that we get here in this country. So vote. Vote. I don't think my box is hooked up to anything in Utah, but I just push it just like it's a Fisher-Price voting booth. I'm like, I'm going to push it again. It just makes me feel better. Okay. What? Better than nothing. Exactly. Okay. So, all right, so let me just tell you what we're going to do with the next set of notes. We're not going to start on them hardcore today, but I will just tell you. So let me introduce you to them big picture, and then tell you about a resource that you might really like. Okay. There's two. I just uploaded these yesterday because I just completely rewrote them so that they make more sense. Hopefully, that was the objective. So after we get past talking about like the protections of the brain, like the dura mater and all of the cranial meninges and then cerebrospinal fluid and blood-brain barrier, the harder, more interesting part is that we are going to start talking about brain structures and functions. And that starts right about here. So one of the reasons that it's a little difficult for students is because there's no real clear boundaries in the brain. Like I mentioned, it's not as easy to see. It's not color-coded once you run into it. It's not. It's just kind of this wrinkly structure. In addition, you may be very good at this, and I'm not. Rotating the brain 3D in space to figure out how things are related to one another is a little difficult. There is an amazing website called 3D Brain. So all you have to do is look up 3D Brain. It's also a free app on your phone, and it allows you to take the brain and rotated in space and put on labels. Rotated in space. See how things are related to one another. Okay, and that's all well and good when you're looking at a whole brain, but importantly when you're having trouble figuring out how things look that are structures within the brain, you can see that's how the corpus callosum looks from a mid-sagittal view. This is how it looks from a dorsal view. This is where it is. And this is just my nerd part. For every single structure that they have mapped in the brain, there is also a case study that like shows you how they understood what that part of the brain does, and some associated functions, disorders about what happens when that thing goes wrong. And then for those of you who are super nerdy like I am, there are like research papers about each part of the brain. So I will kind of use this when I'm trying to get you guys to understand. So the challenge with the brain is figuring out where the part is that you're talking about and then simplifying what the part does. So that's why I rewrote the notes, because I was trying to figure out, okay, what is the clearest way to talk about what it does, assuming that we can find it. So this is called 3D Brain. It's a free app that you can use for iPad or all iOS devices. I don't know if they have other ones. Or it's a website. It was funded by the National Institutes of Health as a part of a huge, huge, huge federal grant for neurophysiology and neuroanatomy. There's a lot more on here too for those of you who are like fascinated by other stuff. It's like also got all of the genetic bases and physiological basis of all of these disorders. But the 3D brain is the part that you would use for anatomy. So I won't test off of it, but I'll use it. And you may want to get comfortable with it so that like when I'm talking, you're trying to figure out where the hell is the amygdala? Oh, there it is. Let's find it. Okay. So we'll start doing all of this stuff next time.